Good afternoon. My name is Jeremy De Silva. I'm an assistant professor of anthropology here at Boston University, and I'm pleased to welcome you to the fourth Dialogues in Biological Anthropology. The dialogues were created by Kay Brown and Matt Cardinal as a mechanism for inviting scholars to the Boston University community to discuss controversial topics in biological anthropology in a non-confrontational, constructive manner. Today's topic is the evolution of the human pelvis. The title is Getting Hip: Babies, Brains, and Bipedality in Human Evolution. The human pelvis is a marvelous example of how natural selection shapes anatomy. Most scholars agree that there have been two strong selection pressures, converting what was once an ape-like pelvis into a more human-like one over the course of the last 6 million years or so. These selective pressures are locomotor and obstetric. We walk differently than the other apes do, and by walking on two legs, our anatomy has altered to maximize the efficiency of this kind of locomotion. But we are not just upright walking primates. We're upright walking primates that give birth to large-bodied, large-brained infants. And selection has also shaped the birth canal. The relative role of these two selective forces is quite contentious and debated in paleoanthropology. And to help answer these questions, we rely quite heavily on the fossil record. The fossil record, however, gives us interesting but conflicting information. There are some who interpret the fossils as evidence that locomotion has been more important in shaping the pelvis. Others have regarded the evidence differently and suggested that obstetrics have been more important. But as with all things, it's probably more complex and much more interesting than just that. And to dive into these complexities and to introduce us to some new fossils that have been found recently that help us understand this question and raise additional questions are two scholars in the field in the subject of the evolution of the human pelvis. We're very, very pleased to welcome Dr. Steve Churchill, who is a professor of evolutionary anthropology at Duke University, and Dr. Karen Rosenberg, a professor of anthropology at the University of Delaware, here to Boston University. We're going to start first with Dr. Churchill, who's going to give a 20-minute talk or so on new fossils from South Africa, from a new species of Australopithecus, Australopithecus sediba. That's going to be followed by a 20-minute presentation by Dr. Rosenberg, who is an expert on the evolution of human childbirth. We'll then return to the panel here and engage in a dialogue and talk about the conflicting information, perhaps, that has been presented and where there might be overlap and consensus about this topic. We'll start first with Dr. Churchill.
Thank you very much, Jeremy, and thank you very much for inviting me to be a part of Getting Hip. I'm very pleased to be here and tell you a little bit about some new fossils from South Africa. So I have here a reconstruction of the pelvis of Lucy. Lucy is a representative of the species Australopithecus afarensis. She lived in East Africa about 3.2 million years ago. And if you compare her pelvis to that of a chimpanzee, you see that there are some remarkable structural differences here. Most notably, her pelvis is shorter from top to bottom, and the iliac blades or hip blades flare out more laterally than they do in the chimpanzee. Now, virtually everybody in paleoanthropology would agree that this has to do with adaptations to bipedal locomotion in the pelvis of Lucy. Lucy walked on two legs, whereas, of course, a chimpanzee is an arboreal climber and a quadruped when on the ground. But if you compare Lucy's pelvis to the pelvis of a modern human, you'll see that there are also some important structural differences between Australopiths on one hand and the genus Homo on the other hand. The pelvis in the genus Homo has more vertically set iliac blades. They're more S-shaped, curved. The birth canal is larger and rounder. The pubic bones are upturned. The pelvis has got more buttressing to it. And the ischia, or what some people call the sit bones, are shorter in our genus, the genus Homo. So what do we make of these differences? Well, that's where opinions diverge a bit in paleoanthropology. And it actually hinges on two models which constitute one of the oldest debates in the field of paleoanthropology. One model is what we might call the arboreality model, and this actually goes back to work in the 1940s by Solly Zuckerman. These days it's most closely associated with three scholars from Stony Brook University, Jack Stern, Randy Sussman, and Bill Jungers. And these guys argue that the total morphological pattern is what matters when you analyze a skeleton. That if you look at Lucy or some other Australopith's total skeleton, you see a lot of primitive features there, features which they think mean that these animals were still doing a lot of climbing. They're terrestrial bipeds to be sure, but they're still doing a lot of climbing in the trees. So the problem for Lucy was that she had a locomotor skeleton which had to meet two very different kinds of demands, climbing and terrestrial bipedalism. So natural selection couldn't really fine-tune the skeleton for either one of those two demands. It was a little bit of a compromise. Because of this, so the model argues, Lucy must have walked with a different kind of gait. She had different kinematics. She walked with a bent hip, bent knee. That's what the BHBK stands for there on the slide. Much like a chimpanzee does when a chimpanzee is walking on two legs. So the proponents of this model would argue that the change to the Homo-like pelvis reflected the relaxation of that arboreal constraint, that we finally have hominids who have come down to the ground full-time, no longer climbing trees, and now selection can fine-tune the pelvis for bipedal locomotion. The alternative model is what we might call the terrestrial bipedality model. This also dates back to the 1940s with the work of Wilfred Legro Clark, but these days it's most closely associated with Owen Lovejoy and Bruce Latimer. And these guys argue that you can forget about the primitive features that you see in the skeleton, that those are just what we call phylogenetic inertia. Those are just features which are still hanging around, but they're not really important to what the animal is doing. If you want to understand what's going on with the animal, you've got to look at the derived features, because this is the focus of selection. The derived features really tell you what this animal is adapting to do. And they argue that when you do that, you see in Lucy and other Australopiths very, very good, competent terrestrial bipeds. They argue that their kinematics were the same as yours or mine, that they were engaging in full striding bipedalism. And furthermore, that the adaptations that occurred in their skeleton for bipedal locomotion kind of ruined them for climbing. So not only were they terrestrial bipeds, they were obligate terrestrial bipeds. They couldn't really climb very well anymore at all. And then finally, these guys argue that some aspects of the locomotor skeleton of the Australopiths, and in particular the pelvis, was better adapted for bipedal locomotion than was the pelvis of the genus Homo. And this last point requires a little bit of exploration, because we wouldn't really expect natural selection to make some sort of functionally important feature worse for the job that it has to do, the function that it has to do. Well, the explanation, according to the proponents of this model, has to do with brain size expansion in the genus Homo. They argue that as brains got bigger in our genus, it forced an enlargement of the birth canal to accommodate the passage of these large brain babies during birth. And given some constraints on the size and shape of the pelvis, that required architectural changes in the overall structure of the pelvis, which kind of ruined the efficiency of the pelvis or worsened the efficiency of the pelvis for bipedal locomotion. So if you look at this quote by Owen Lovejoy, he would basically argue that the differences between the pelvis of Australopiths and Homo are really about brain size and encephalization, not about locomotion. And in fact, in a series of papers, Lovejoy laid out exactly how those changes occurred. He argued that there were three major architectural changes to the pelvis to accommodate bigger brain babies. These included a relative increase in the sagittal diameter of the pelvis, an absolute increase in the transverse diameter of the pelvic inlet. So these two things increase the size of the birth canal, and an upward rotation of the pubis and a downward rotation of the ischium to provide a little bit more room in the birth canal. And then, because of developmental relationships within the pelvis, there were secondary changes that came about because of those primary architectural changes. These include a reduction in the relative length of the superior pubic ramus, more vertically and less flared iliac blades, a more posterior and robust iliac pillar, greater robusticity of the ilium, and reduced distance from the acetabulum to the ischial tuberosity, a shortening of the ischium. So, we're sort of left with this question: what do the differences between the pelvis of something like Lucy and the pelvis of a member of the genus Homo mean? Is it about locomotion, or is it about obstetrics and encephalization? And there are two new skeletons from South Africa, from a site just outside of Johannesburg, the site of Malapa, which I think shed some light on this long-standing debate. These skeletons are dated to about 1.98 million years. We have the partial skeleton of a subadult male, MH1. He is the type specimen of a new species, Australopithecus sediba. And we also have the partial skeleton of an adult female, MH2. She is the paratype specimen of this new species. And as you can see, both of them preserve some elements of the hip. We announced the new species about a year ago in April of 2010, and at that time we only had portions of the pelvis of the subadult male, MH1. For those of you who are following along the webcast, looking at the PowerPoint slides, I have to tell you I've combined two slides into one, so I hope that isn't too confusing. You can figure it out. But anyway, this is what we had when we went to press. We had portions of the right iliac blade of MH1 and the left iliac blade and left ischium of that individual. But that was enough for us to see that there were some derived features here in Australopithecus sediba that you only find in the genus Homo and which are unknown in earlier Australopiths, like Lucy, that's AL288-1, or STS14, which is a representative of Australopithecus africanus from South Africa. So we're seeing these derived features, which you only see in the genus Homo, but the adult brain size of Australopithecus sediba seems to fall well within the range, and in fact on the small end of the brain size of Australopiths generally. So this suggests that something is a little bit wrong with the obstetric model, because we're seeing these derived features in the absence of brain size expansion in this species. Now, people could say, well, you only had the pelvis of a juvenile male. How do we know what a female would look like or what an adult would look like? Well, about a month after we went to press with the announcement, we actually recovered portions of the pelvis of the adult female. We already had her right pubic bone, but we found her right iliac blade, we found a portion of her sacrum, and importantly, we found a little linking piece which links her pubic bone to the iliac blade. This material gave us sort of enough to work with that we could very confidently reconstruct half of the pelvis, which could then be mirror imaged to get a look at the total pelvic architecture. So here's what the hemipelvis or half pelvis looked like. You can see that a portion of the anterior part of the iliac blade is missing. That had to later be reconstructed, but fortunately we had the existing curves of the iliac blade to guide us in that part of the reconstruction. And here's what the reconstruction looks like. The adult female MH2 is on the right in two views. In the center is a virtual reconstruction of STS14, representing Australopithecus africanus. And on the far left is our reconstruction of MH1. So we also reconstructed his pelvis, although we had less to work with there, and accordingly we don't have as much confidence in that reconstruction. But you can see that there are some major architectural differences here between the pelvis of MH2 and the pelvis of STS14. The iliac blades are somewhat more vertically set. The superior pubic rami are upwardly rotated. Here are our two pelvis relative to a reconstruction of Lucy's pelvis. I should point out that this reconstruction and the reconstruction I was showing you a minute ago were done by Peter Schmid. Owen Lovejoy has also reconstructed this pelvis, and his reconstruction is a bit more modern looking, although not markedly so. So, a couple of things that I want to point out about the overall pelvic architecture in Australopithecus sediba. First off, if you look at the bottom three rows of this table, the left-hand column is the dimensions of MH2. The next column to the right is Australopithecus africanus, and then Lucy, Australopithecus afarensis. And then the last column, which doesn't have a picture above it, represents a specimen which has been attributed to Homo erectus from East Africa from a site called Gona. And if you look at the last three lines, those are the dimensions of the pelvic inlet. First the transverse dimension and then the sagittal dimension. And what you can see is that in the absolute dimensions, MH2 is not that different from STS14 and not very different from Lucy. So we're not really seeing an expanded birth canal, but you can see that the shape is a bit different, that the ratio of those two measures shows that MH2 has got a more sagittally expanded birth canal. The same, however, is true of STS-14, and it's really Lucy which has got a transversely very, very wide pelvis. And then in the top three rows are biiliac breadth and biacetabular diameter. That's a measure of the total width of the hip blades versus the distance between the hip joints. And if you look at the ratio there, you see that we have a very low ratio in MH2, and that indicates, again, the more vertical orientation of the iliac blades in this specimen relative to the Australopiths. You see that here as well. This is the iliac angle, the angle between the iliac blade and the superior pubic ramus. You can see that in the genus Homo, represented by the little box plot, that that angle tends to be a bit more closed. Australopiths like STS-14 have a much higher angle, and there's MH2 falling sort of in the upper end of the range of modern humans. The blue dot represents a Neanderthal, Kabara 2. And again, this is because both the iliac blades are more vertically oriented and because the superior pubic ramus is upwardly rotated. Now, with respect to the iliac pillar, we're getting kind of a mixed signal here from the material from Sediba. MH1 has got a distinct iliac pillar. It's not very large, but it is a distinct iliac pillar, and that's like what you see in the genus Homo. MH2, on the other hand, has got more of the Australopith configuration of a very indistinct and anteriorly positioned pillar. So we're kind of getting a mixed signal on this one trait. With respect to the robusticity of the iliac body, this ratio is simply a measure of the stoutness of the weight transfer region between the sacroiliac joint and the hip joint. And what you can see is that Australopiths tend to be very gracile down there. They tend to have a very long and not very thick weight transfer region. In the genus Homo, both archaic or Pleistocene members of the genus Homo and modern members of the genus Homo, you see a much thicker, stouter weight transfer region. We see the same thing in both MH1 and MH2. So again, this is a derived characteristic in the Malapa hominids. And then the last feature that I'm going to talk about is the length of the ischium. This is a measure of the relative size of the distance between the acetabulum and the ischial tuberosity. And what you can see is that in the genus Homo, again, whether we're looking at archaic or modern humans, the ischium is very short, and that sulcus between the acetabulum and the ischial tuberosity is very, very narrow. MH1 has got the same configuration. In the Australopiths, you tend to have a very, very large distance there. This next picture just shows you this in a couple of specimens. So on the left, we've got two Australopiths, Lucy and another specimen from Hadar, and you can see the great distance between the acetabulum and the ischial tuberosity. On the right, we've got MH1. These are all scaled to the same size, and even though MH1 is a little bit of a larger individual, you can see how narrow that sulcus is between those two structures. Now again, you might say, well, MH1 is a juvenile, and maybe the ischium would continue to grow with the last few years of development that he has left. But if you look down at the bottom of the slide, this is a juvenile specimen from Makapansgat representing Australopithecus africanus. This is an individual who is developmentally much younger than MH1, a very much smaller individual, and already that sulcus between those two structures is absolutely larger than we see in MH1. And this suggests to us that this feature is established very early in development for the Australopiths. So going back to these changes which Owen Lovejoy has argued represent the results of encephalization and having to have a larger birth canal, we're seeing some of these architectural changes already in Australopithecus sediba, despite the fact that the brain size isn't any larger than any other Australopith. We are seeing that rounder shape, that greater sagittal dimension to the birth canal, and we're seeing an upward rotation of the pubis. We're seeing some of the secondary changes. We're seeing more vertical and less laterally flared iliac blades. We have a more pronounced iliac pillar in one of the specimens. We have greater robusticity of the ilium, and we have a reduced distance from the acetabulum to the ischial tuberosity. So these things all suggest that while obstetrics was important, I'm not going to say that obstetrics wasn't important to the evolution of the human pelvis. I think it was probably very important in establishing the kind of sexual dimorphism that we see between males and females today. But it doesn't explain all of the differences between the pelvis of Australopiths and the pelvis of the genus Homo. Thank you.
Thank you very much, Steve, for speaking about those really extraordinary new fossils. Next, we're going to hear from Dr. Karen Rosenberg of the University of Delaware, who's going to present a slightly alternative view in interpreting the human fossil record. Dr. Rosenberg.
Thank you, Jerry, and thank you very much to Kay and Matt for inviting me. Childbirth is a time in the human life history when it's quite easy to understand how natural selection acts on human form, both of the mother's pelvis and of the infant itself. Nevertheless, when I started graduate school 30 years ago, very few paleoanthropologists had really considered the evolution of human childbirth as a topic of study. I remember that one of my professors said early on when I was studying, however, that Australopithecines were well-adapted bipeds, and that all the changes in the pelvis that we see since then reflected changes to accommodate an increasingly larger brain, and that all of those changes represented had negative consequences for bipedalism. So I think we can see, especially from what Steve was talking about, that simple view is clearly not, can't be held any longer. So childbirth in humans is risky, complicated, and painful compared to the way it is in other animals, and in particular compared to other primates. In humans, the baby generally passes through the mother's birth canal, undergoing a series of rotations to accommodate itself to the shape of the birth canal, which changes in cross-sectional shape as you pass from the inlet to the outlet. And these are the cardinal movements of labor that obstetricians recognize, and that is the typical way that a human infant passes through the birth canal as it emerges from the mother's body during childbirth. You can see in the little corner of the picture, what you're looking at is the baby's head. If you were the obstetrician with X-ray vision, or the midwife with X-ray vision, you would see the baby's head. And I've put a little red arrow on the baby's head to illustrate the longest axis of the baby's head, because what's interesting in humans is that because there's such a tight fit between the mother's birth canal and the baby's head has to align itself along the longest axis of the mother's birth canal. And that's why you see that arrow changing as it passes through. So typically the baby's head enters pointing to the side or pointing obliquely, and then it passes through rotating so that it typically emerges in a position that obstetricians call occiput anterior, with the back of the baby's head, the occiput of the baby's head, towards the front or anterior of the mother's body. And here you see the baby's head on the right of the picture in red, rotating as it passes through the mother's birth canal and emerging in that occiput anterior position. Because the baby emerges in that position, humans benefit a lot from having somebody there to help the baby and to help the mother during the process of childbirth. And in societies around the world, it is invariably the norm that birth takes place assisted, that someone is there to help the mother and to help the baby during childbirth. The figure on the left shows the three planes of the birth canal that obstetricians define: the inlet, the midplane, and the outlet, in side view at the top and in front view at the bottom. And I'll be referring to those planes in a few minutes. So for humans, because of the complicated way that we give birth, because of the way that the baby emerges, and because of the tight constraints between the size of the baby's head and the size of the mother's birth canal, humans benefit from assistance during birth. And here you can see the midwife receiving the baby as it emerges from the mother's body. The situation in other primates is quite different. This is a picture which shows infant cranial size relative to maternal inlet breadth. So this is the top of the birth canal. And you can see monkeys and then gibbons in the top of the picture, and great apes and humans on the bottom. And the picture represents the black oval represents the size of the baby's head, and they're all scaled to the same size. And then the outer oval represents the size of the mother's birth canal at the inlet. And you can see that for the great apes, Pongo, Pan, and Gorilla, the baby's head is much smaller than the size of the mother's birth canal, and there's quite a lot of room to spare. But for the smaller bodied monkeys and gibbon, there's quite a tight fit between the baby's head and the mother's birth canal. And this is reflected in the observations that we have of primates giving birth, that it seems to take more time, it seems to be more difficult for monkeys than it is for apes. For humans, Homo over there on the far right, lower picture, you can see that the length of the baby's head is actually longer on average than the front to back dimension of the mother's birth canal. And that's why human babies typically enter the birth canal facing to the side. Here are just some numbers to show that. These are the numbers showing the infant head dimension as a percentage of maternal pelvic inlet dimension. So for spider monkeys, it's 74%. For macaques, 98%. Gibbons, small-bodied apes, it's still quite high at 82%. But for chimpanzees, the baby's head is just over half the size of the mother's birth canal. So there's a lot of room to spare. And for humans, the baby's head is actually larger, 110% the size of the front to back dimension of the mother's pelvis, so that it enters facing to the side because the transverse diameter is bigger. And here's a drawing of a monkey on the left and a human on the right, shown from the side, of the baby's head entering the birth canal. And you can see that the human baby is entering facing to the side, and then as it passes through the midplane and outlet, it's going to rotate facing towards the back. And the monkey enters the birth canal facing forward with its head down in that position. And then it typically emerges with the face forward rather than in the occiput anterior position. But there's been some recent work in chimpanzees suggesting that they sometimes give birth at least sometimes give birth in occiput anterior position as well. For most mammals, and certainly for most primates, birth is typically a solitary activity. So most mammals give birth as far away from other members of their group as they can. This is some drawings of a Japanese macaque giving birth, and they try to find some kind of isolation. You can see that this macaque mother is able to assist the baby as it emerges from her birth canal, something that human mothers can't do as easily. And actually, monkey babies are able to assist somewhat in their own delivery because they have the strength to grab onto their mother's fur, and their mothers have fur, and they often actually work their way up the mother's body on their own. So most mammals give birth alone. This is a photograph of a non-human primate on the left and a human on the right, just to reiterate that human primate birth canal is a shallow, bony ring in which the inlet, the midplane, and the outlet are all long from front to back, and humans have a very deep curved tube of the birth canal, which has a changing cross-sectional shape. So it's widest at the top, then longest from front to back, and then approximately round at the outlet. So where did this come from? We used to talk a lot about the obstetrical dilemma, which was a way of describing the fact that the human pelvis was an adaptation to both bipedalism and also to the birth of large-brained babies. And in the simple-minded view that I described before, we understood that these were the two selective factors shaping the evolution of the human pelvis, mediated somewhat by the evolution of helpless newborns. So because we could change the point in development at which human babies were born, that was one way of mediating the two constraints of bipedalism and large brains. So what do we know today? I'm sorry, one more thing. This is a graph of brain growth against body weight growth. And this is just to illustrate the point that human babies are born at an early stage in their development relative to brain growth. So you can see in this graph that brain growth is very fast in utero, and then it levels off. And most primates give birth at about that inflection point. So chimpanzees give birth at about the time when brain growth slows down. But humans continue to grow at essentially fetal rates of growth for some period of time after birth. And so they're more helpless at the time of birth. This table shows some numbers to illustrate that. You can see this is newborn brain size as a percentage of adult brain size. In the monkeys, the numbers are in the 50s or higher. In apes, in the 40s. And humans are born with the baby's brain size being about 29% of adult brain size. So they still have a lot of growing to do after the time that they're born, and they're very helpless at the time of birth. And here you can see a helpless newborn human again on the left, and a newborn gorilla on the right. And you can see this comparison, I think, is pretty remarkable. And this is what led Ashley Montagu to call humans exterogestate fetuses. Ashley Montagu argued that humans could be born at a very early stage in their development because we have culture to buffer us from the environment. And he argued that in the early months of babies' lives, they were essentially being buffered by a kind of cultural womb, and by things like this, by cradle boards, today by incubators, by being carried inside of their mother's clothing. So they were somewhat buffered from the environment. And that we're able to do that because we're cultural animals. So you can see two places here that human culture is able to intervene. One is that we're able to give birth to these large-brained babies that are born in this complicated way because we can assist each other during birth. And then we're able to provide protection and buffering to newborn babies through culture as well. Okay, so how did this evolve? This is a drawing of chimpanzee pelvis at the top, Australopithecus in the middle, and Homo at the bottom. Owen Lovejoy and Bob Tague used the anatomy of Lucy about 25 years ago to reconstruct the birth process in Australopithecines. They argued, this is now again the sort of X-ray vision view of the birth canal, if you were looking into the birth canal as the obstetrician or midwife. And you can see chimpanzees on the left, Australopithecines in the middle, and Homo on the right. In chimpanzees and in other primates as well, the baby seems to have plenty of room to spare in the birth canal. We know now that the babies do rotate somewhat, but not according to a typical kind of pattern. In Homo, the baby enters the birth canal facing to the side and then rotates to emerge, typically in the occiput anterior position. That's not the only way that babies emerge from the mother's birth canal, but it's by far the most common, and importantly, it's the one that's associated with the lowest risks. Tague and Lovejoy argued that Australopithecines, because they had a birth canal which was wide, or what obstetricians call platypeloid, flat and wide, at the inlet, at the midplane, and the outlet, they argued that Australopithecine babies would have entered the birth canal facing to the side, like humans, and that they not only didn't need to turn to negotiate the rest of the passage, but that they could not turn, that there wasn't enough room to spare, or to turn, and that they emerged from the birth canal in that transverse orientation. This means that Australopithecines would have had a mechanism of birth completely different from anything that we see today, which shouldn't be too surprising because they had a combination of a bipedal form of locomotion and a small brain, unlike anything that we see today. This is the same picture, basically, that Steve showed you of Australopithecus sediba, Australopithecus africanus, the two sediba ones on the right and the left. And he argued that this pelvis shows some changes in the direction of Homo, which can't be explained because of enlarged brain size, because this is still a small-brained creature. Previously, our understanding of the evolution of childbirth in early Homo depended on this specimen, the Neonatal specimen, which was discovered 20 or 30 years ago and described by Alan Walker and Chris Ruff. And you can see that the pelvis there is quite fragmentary. In addition, it was also a male specimen. It was also a juvenile specimen. And again, it was fragmentary. So we had to make a lot of assumptions to go from this juvenile male fragmentary specimen to understanding what Homo erectus childbirth might have been like. They argued that, that rotational birth did not occur in this early Homo. Today we have this new specimen from Gona, which Steve also talked about, and this picture shows Australopithecus afarensis, Homo erectus, and Homo sapiens on the right. And the Gona specimen shows some expansion in the anterior-posterior direction, front to back, as Steve mentioned, and that may be related to increasing brain size. This is another picture of the same thing, showing that the Gona specimen would have been able to give birth to a larger-brained baby than the earlier Australopithecines, and in this case, almost within the modern range that is indicated on this diagram. This graph shows the Gona specimen compared to modern humans. So the red funny circle is the Gona specimen, and we have the inlet on the left and the midplane on the right, and longer from front to back, birth canals are on the upper left, and flatter is to the lower right. And the squares and circles represent modern humans. And you can see that the Gona specimen is within the range just at the inlet, somewhat outside the range at the midplane. So it's expanded, but not as much as modern humans. Following the Homo erectus, we have a bunch of nice specimens that I just wanted to quickly mention. In the latter period of human evolution, this is the Kabara specimen from Israel, a Neanderthal, which has a very modern pelvis with respect to the birth canal. We have another Neanderthal from Taboon, which has been reconstructed as showing evidence of rotational birth. These are humans giving birth to very large-brained babies, relatively recently in time in human evolution compared to the other material I've been talking about. And this is the Jinyushan specimen, which I've been working on, a specimen from about 250,000 years ago from far northeastern China. A very large-bodied individual which shows all of the adaptations of the birth canal that indicate rotational birth of large-brained babies. And this is a picture of the birth canal inlet of the Jinyushan specimen, showing this anterior-posterior expansion. If you remember what the Australopithecines looked like, this is much longer from front to back. And that indicates almost certainly that there was rotational birth. Okay, so just to review what we know about birth canal shape in the human fossil record. This shows the inlet, the midplane, and the outlet. And an index which is below 100 is flat, or platypeloid. An index above 100 is elongated from front to back, and 100 would be approximately round. So you can see that modern humans, and there's two populations of modern humans over there on the right, are flat at the inlet, long from front to back, and then pretty close to round at the outlet. And that's what I meant at the beginning when I talked about a birth canal of changing cross-sectional shape. The Australopithecines, Lucy and Steve's Sediba specimen here, Lucy is flat at the inlet, flat at the midplane, flat at the outlet, which was why Tegan Lovejoy reconstructed birth as non-rotational. And in the Sediba specimen, it's flat at the inlet and then closer to round at the midplane and the outlet. In Homo erectus, we have still the same flat inlet, which seems to be the primitive condition for humans, something approximately round at the midplane, and we weren't able to measure the outlet. And then for the archaic humans, we have something much more like the modern human pattern. An inlet, birth canal which is flat at the inlet, long or round in the case of Taboon at the midplane, and then the outlet measurements are a little bit less reliable, but they seem to show expansion there as well. Steve said that, this is a quote from the paper that Steve wrote, where he said, "These derived features in the Sediba specimen appear in a species with a small adult brain size, suggesting that the birthing of larger brained babies was not driving the evolution of the pelvis at this time." We agree on this. However, I want to argue that birth is about more than just the head. The head is important, certainly, and in many cases, especially in the later periods of human evolution, it may be the most important factor, but it's not the only one. One often overlooked factor is the width of the shoulders. This is a picture of a monkey and an ape, and you can see that apes, like humans, have a broad, rigid thorax. Monkeys have a narrower thorax, which is longer in the anteroposterior dimension. In humans, shoulder dystocia, or obstruction of the shoulders, is a common source of difficulty during childbirth, and it's often alleviated by manipulations that obstetricians and midwives do to make a little bit of extra space. But the shoulders represent, although the head is the largest thing that has to pass through the birth canal, the shoulders can also become obstructed. So I think that it's important to consider that Australopithecines, although they were small-brained, were broad-shouldered, like humans and the other apes. Another thing that's often overlooked that Jerry De Silva has worked on is neonatal body size. This little table shows newborn size as a percentage of mother's size. So for gorillas and chimpanzees, babies are much smaller in body size compared to the size of their mother than humans. Gorillas are 2.7% of their mother's body size, and chimps 3.3%, and humans on average are over 6% of the size of their mothers. So when you think about humans giving birth to helpless offspring, remember that another thing about those helpless offspring is that they're very large. No wonder they take so much care. This is also a figure from Jerry's work showing the infant-mother mass ratio for humans, some apes, and fossil humans. And you can see that for chimpanzees and gorillas, as I said before, the infants are very small relative to the sizes of their mothers. For humans, infants are larger relative to the size of their mother, and that seems to be something that's been true for a long time in human evolution. So we've probably been giving birth to large babies longer than we've been giving birth to large-brained babies. And I think it's important to consider overall body size of infants as well as brain size. So, in conclusion, human birth anatomy is constrained by bipedalism and encephalization for sure, but not only by those phenomena. Secondly, we need to consider neonatal body size, shoulder breadth, and infant maturity at birth in understanding the evolution of human birth and in understanding the evolution of the human pelvis. The obstetric challenge on the human pelvis comes not only from the increasingly large human brain, but also from the changing mechanism of birth, the changing way that the baby passes through the birth canal, as well as other mechanical and developmental constraints, always mediated by culture. Thank you.
Thank you very much, Karen and Steve, for fascinating talks. What we're going to do now is we're going to gather to my left here and start answering some of the questions that you submitted in watching the webcast via the BU Dialogues Facebook page or via email. And I want to start with a question about alternative interpretations of the pelvis, not just related to obstetrics, but also locomotion. So we'll... So one of the questions that we received from students watching this was the role that the size of the gut has played in the evolution of the pelvis. That in addition to obstetric constraints and locomotion, what about the fact that the gut is actually quite large in chimpanzees and gorillas? And what do we know about the diet of Australopithecines that might be influencing how the gut is shaping pelvic anatomy?
That's a really good question, and that's something I've been thinking about a lot lately, as a matter of fact. And, you know, I have to tell you, I'm not entirely sure what the effect of gut size is, but you have to imagine, you know, our dietary reconstruction suggests that the Australopiths were eating a largely vegetable-based diet, probably a highly fibrous diet, not dissimilar to what chimps are eating, a good bit of fruit in there and whatnot, but a highly fibrous diet, so they probably had a very long gut. And so it's quite possible that as a biped with a large gut, that's also affecting pelvic morphology. And particularly, this may have posed some problems for controlling trunk rotation during bipedal locomotion. You know, the trunk tends to swing away from the swing phase leg, and we control that by counter-swinging the opposite arm. But if you've got this big gut sticking out in front of you, then that, you know, you got some inertia from that gut twisting your trunk. So it's possible that some of that iliac flare that we're seeing in the Australopiths is about some of the abdominal oblique muscles and whatnot having to be better advantage to control that trunk rotation. But beyond that, I'm not really sure what, how else this might have affected bipedalism.
Yeah. Now, what about, I mean, would we expect, therefore, in the evolution of genus Homo, which, if they're eating more high-quality foods and eating more meat, as has been reconstructed, that you would see a corresponding reduction in the iliac flare? You'd see the same kind of changes that some have interpreted as changes related to an increase in brain size. It's almost as though all these variables are happening all at the same time in that transition from Australopithecus to Homo. How do we separate them? Or can we?
Yeah. Karen?
Yeah. No, I think it's actually something that's really been overlooked, that people have suggested for a long time that supporting the gut was an important function of the pelvis to look into, but there hasn't been as much work on it as there should have been.
Yeah.
Yeah, you know, there's one thing when you see Homo erectus, or what some people call Homo ergaster, early African Homo erectus, on the scene, you see a whole package of changes. You see the fully established Homo-like pelvis, but you also see longer limbs, you see a larger body, and all of those things seem to suggest that there are a bunch of interrelated changes going on. You know, along with that are smaller teeth and smaller chewing muscles and stone tool use and more manipulative hands and all that, and it does suggest that there's higher quality diet, probably more tool-based extractive foraging, and maybe that involves a greater amount of ranging behavior. They're incorporating more carcasses into their diet, and they're having to get out in open landscape to find those. They're ranging farther. But you're right, they're all sort of changing at once, and it's very, very difficult to tease apart what the selective agents are, and, you know, whether you know, is the pelvis about locomotion, or is it about the bigger brain that we're seeing all of a sudden, or is it about a smaller gut, which may have to do with a higher quality diet? And I think that's a challenge to us. Mm-hmm. I don't have a ready explanation of how we figure something like that out, but I think that's the exciting challenge. You know.
So, Karen, there's a question directed more towards you about rotational birth. And there's a recent paper suggesting, and you alluded to it as well, that chimpanzees actually have rotational birth. So if that's the case, if chimps have rotational birth and humans have rotational birth, does that mean that the arguments that have been going on about whether Australopithecines or Homo erectus or Neanderthal had rotational birth, are those now irrelevant? Can we just assume everyone had rotational birth?
I think for a long time we've been assuming that the occiput anterior position in which human babies are born was a unique derived feature of humans. And I think we have to question that now because of this evidence that these chimpanzees gave birth, at least some chimpanzees gave birth in a occiput anterior position. Remember that the chimpanzee head is still much smaller than the size of the mother's birth canal, and in fact it can rotate quite a bit because there's really very little in the way of constraints, bony constraints, the way we have in humans. So I think we need to sort of reconsider some of those old assumptions that we were making before in light of the chimpanzee data. One thing I would say is that even if Australopithecines gave birth in a non-rotational way as far as the head was concerned, they wouldn't have been able to give birth in a non-rotational way as far as the shoulders were concerned, because they were, again, small-brained but presumably broad-shouldered human ancestors with a very flat pelvis at all three pelvic planes.
So one of the questions, again, that a student has asked is, it seems so obvious that we would know how a chimp gives birth. So why has it taken so long to figure this out?
So primates, because they give birth on their own, without attendants, when they give birth in the wild, they try to get away from conspecifics, they try to get away from predators, and they try to get away from primatologists. So it's very unusual, almost non-existent, for primatologists to witness birth in the wild. We've witnessed birth in labs and in zoos a fair bit, but in the wild we haven't seen too much. So we really don't know as much about wild animals giving birth as we'd like to, and it's very difficult data to collect.
Mm-hmm. Mm-hmm. So well, I guess, why does it matter? Why does the rotational birth actually matter in terms of reconstructing, say, social behavior or something like that in Australopithecus or genus Homo? Would that necessarily make it a more difficult birth?
Yeah, I think it would. I think rotational birth and persistent occiput anterior birth and a lot of the other constraints, that the fact that we're giving birth to such helpless offspring, the fact that the shoulders can easily become impacted, and when the shoulders are impacted, what often happens is injury to the brachial plexus, which can lead to paralysis or a kind of palsy. And those are things that can all be alleviated by the presence of an assistant. And I don't mean necessarily an obstetrician in a high-tech environment. I mean someone who has observed the birth process before and knows what they're doing and knows how to use some of these maneuvers, or move the umbilical cord from around the baby's neck, or clear the baby's breathing passage, or support its neck as it emerges, things like that, which humans benefit from in a way that apes don't, because of the way that they're born and because of the more developed condition of the newborn.
Mm-hmm. Okay.
Well, one thing that I think is really interesting, which I came to appreciate reading Karen's statement, is, humans are cooperative breeders. And most of that we tend to think about in terms of allocare, attention given to offspring by a non-relative, in terms of caloric support, so that members of a social group who are actively reproducing may get more calories from people who don't have dependent children. But, Karen's stuff really points out, it begins with the birth process, that we have cooperators who are there, basically helping out from moment one. And I think that's a really interesting sort of human feature.
Mm-hmm. Mm-hmm.
Yeah, and I think it says something about the relationships that human females form within the group and with each other. Because typically in most societies it would be mothers or sisters or close female relatives who are helping in the birth process.
How far back do you think that goes? Or is that something that's really difficult to answer at this time?
I don't know. I mean, I feel pretty confident that by the time we have big brains, 250,000 years ago, I think we have assisted birth. I guess when I look at Homo erectus, I think it's likely. And I'm not sure about Australopithecines. I think it's possible that it sort of depends on our reevaluation of the mechanism of birth, but I think it's possible that they would have benefited from assistance too.
Mm-hmm. So I'll shift gears now and ask a question that was directed towards Steve regarding fossils and the fragmentary nature of the pelvic fossil record. Mm. Now it seems as though just about every fossil pelvis we have is distorted, reconstructed. So how do we know we're right on these reconstructions?
Well, we don't know we're right. So this is a problem. I mean, we're talking about a portion of the skeleton which is a bit more prone to breakage and taphonomic damage, over the many millennia that these things are sitting in the earth. And they do have to be reconstructed. And the problem with reconstruction, and this affects everybody who does reconstruction, I'm as guilty of it as anyone else, is that our preconceptions about the way these things look enter into the decisions that we make about how parts go together and all the million little decisions that go into fitting these things back together. But the healthy thing about science is that you know, we produce a reconstruction and 9 other people will challenge it, you know, and then a few of those 9 other people will create their own reconstructions. And so the whole process is actually, I think, a good one of sort of rational skepticism about these reconstructions and what they mean. Mm-hmm. And I think it's just an inevitable part of a science that deals with fragmentary fossils.
Yeah, yeah. Now, are there alternative reconstructions of Sediba in the works, or have you attempted other ways of putting that specimen together?
We feel fairly confident about this reconstruction because we were lucky to have parts which make fairly good contact. There were a couple of places where decisions had to be made, and we felt that we made defensible decisions. Maybe perhaps not the correct ones, but we can explain why we did it, mm-hmm. and defend why we chose to do things the way that we did. So at this point, we feel satisfied. Now, anyone else is welcome to do their own reconstruction, and we'll probably even supply them with the materials to do so. But the great thing about Sediba is that probably more material is going to come out of the ground. And so it's quite possible that our reconstruction will be challenged by our own fossils, you know, that we'll find the other half of her pelvis and we'll go, wow, the iliac blade really kind of didn't look like the way we reconstructed it.
Sure.
You know, so it's a good situation to be in, though.
Yeah.
But I would say we don't know we're right. We know that we have the best we can do at the moment that's consistent with the most other pieces of data that we have, and that it's a self-correcting process.
Sure, sure, until new evidence emerges.
Yeah.
Right. Another question that was asked by several students, we're interested in how much we can use modern humans as a model for understanding these 2 selective pressures, because obviously females are the only ones that give birth. And so if you look at the sexual dimorphism in the modern human and modern female, human male and modern human female, does that give any insight into this question of locomotor versus obstetric differences? Do men and women walk differently because our pelvis is shaped differently?
Yes. I think I think that the differences that you see in the anatomy of the pelvis and in locomotion in humans are a reflection of the fact that females are under direct selection because of obstetrics and males aren't. But selection from obstetrics does affect males indirectly as well. So
why?
Because half of the babies that are born are male, right? And because males have mothers and daughters that give birth, with whom they share genetic information.
But I often have students ask me if selection is operating on the female pelvis, why is the human male pelvis also evolving in the same direction?
Yeah, exactly.
And I think the answer to that is that the genes which probably control those developmental events are autosomes. They're not the sex chromosomes, on the sex chromosomes. And so that the males are just sort of brought along as selection is operating on the female pelvis.
Do we see that kind of dimorphism early in development, or is it something that happens much later, you know, during puberty or later than that?
Well, I would say that some of the basic aspects of our pelvis occur very early in development, but real sexual dimorphism, the real differences between males and females, only seem to develop under the influence of the sex steroids after puberty.
And one final question that was asked is whether the modern human pelvis, as we see it now, has reached this perfect compromise between birth and locomotion, and if there are any additional selection pressures that might be operating.
It doesn't seem like a perfect system to me.
No.
No, it seems like, to me, it seems like a compromise that just barely works, actually. So we're adapting to conflicting constraints, and we're able to accommodate them in a very precarious balance.
Great. Well, I want to thank you two very, very much for such an interesting and stimulating discussion. Those of you who've watched the webcast and are interested in coming to the 9th floor of the Photonics Center at 4 o'clock, we will continue our discussion, and you'll be welcome to come and direct your questions right at Steve and Karen yourselves. We look forward to seeing you. Thank you very much.